All right, guys. Today we're going to go over sensory pathways and somatic nervous system. All right. So to start here, there's a similar picture in your book here, just an overview of how our sensory and motor pathways work, showing we have an arriving stimulus that causes a depolarization in the sensory receptor, which then propagates an action potential into the central nervous system where it's processed, and then we send signals down through the motor pathways to involuntary and voluntary muscles as a response. So we'll go through the specifics of the pathways, talking about the receptors, specific pathways, sensory pathways up to the brain, and then specific motor pathways down. So sensory pathways, again, that's afferent division. Okay, so that's information coming in towards the central nervous system. Okay, it starts at the sensory receptors and then goes through specific pathways up to the central nervous system. We have the sensory receptors, and then when they're stimulated and they convert that stimulus into an action potential, that's something called transduction. Okay, so that stimulation of the sensory receptor then converts into the impulse, the electrical impulse of the action potential, which then is carried to the central nervous system. So that's a process called transduction, and then we sense that, and we have a sensation when the information arrives in the brain, and then our conscious awareness of that is perception. Okay, so the stimulus gets to our brain, and then we become aware of it and realize what it is, and that conscious awareness is called perception. Okay. And then we have, so that's our sensory pathways, and then we have motor pathways, which are control the peripheral skeletal muscles and visceral smooth muscles, things like that. And that is our efferent division, going from the central nervous system to the effector organs. So we're going to talk about some receptors, some terminology related to that. So receptor specificity. This refers to the fact that we have different types of receptors that can sense different things. Okay, not all the receptors sense pressure. Not all the receptors sense taste or smell. So we have different receptors that are sensitive to different things. So our taste receptors sense taste. They're sensitive to these dissolved chemicals, so they can sense taste, but they don't sense pressure. Okay, so there's differences among the receptor types, and that's called receptor specificity. Okay. And then we have free nerve endings. We'll talk about free nerve endings a little bit too, but in here it's referring to the receptive field. Okay, the receptive field is the area monitored by a single receptor cell. Okay, so the free nerve endings here, you can see one receptive field here and another one here. So those are dendrite extensions, okay, that are sensitive to a specific area. Okay, so the larger the receptive field here, the poorer the ability to localize the stimulus. So when you have larger receptive fields, like say on your back or on your leg, it's harder to specify exactly where the stimulus is, as opposed to if you have sensation on your fingertips or on your hand or on your tongue or something, you can specify more clearly where the sensation or the stimulus is. We talked about transduction, changing that stimulus of the receptor to an actual action potential. And then receptor potential. This is just another term for like the membrane potential of a receptor. Okay. So we have like in our nerve cells, the resting membrane potential is at -70. Okay, so the receptor potential is just the membrane potential of the specific receptor we're talking about. And then a generator potential is a depolarizing receptor that creates that graded potential or action potential. Okay. Label line. So this is another concept that label line is a specific link between peripheral receptor and cortical neuron. So there's a line basically from the receptor to a specific area in the cortex. Okay, so when this is stimulated, anywhere along this pathway, you have this a sense of the stimulus. Okay, so the label line consists of axons carrying information about one modality. Okay, so one type of stimulus. So say pressure. Okay, you have the sense of pressure. Okay, those axons carry that sense, that action potential about the pressure to a specific area in the central nervous system. Okay, and if this label line, this line of the neurons of axons are stimulated anywhere on that pathway, you have the stimulus, the sensation in your brain that something is occurring, whether that's actually true or false. Okay, this is like the phantom limb. Okay, when you don't have part of your arm or leg, if it's been surgically removed, if you still stimulate that label line along that neural track, you can still sense that something is touching your foot, even though if you don't have a foot there. So you have that sensation that it's there where it's not really there. So that's why we say you can't really differentiate between true or false sensations if this label line is activated. Okay. So we have two different types of receptors. You have one that's always active, a tonic receptor, and one that's usually not active. Okay, and this helps to convey characteristics of a stimulus. Okay, like the frequency and pattern of these receptor stimulations can allow you to sense the stimulus, the strength of it, the duration, variation in it. So tonic receptors are always active, okay, and they increase or decrease in frequency. And this increase and decrease in frequency of action potentials directly reflects the increase or decrease in the stimulus. So you have more stimulus, you have a higher increase in these receptor stimulation, this receptor action potential, and therefore you feel that you have a sensation of more stimulation. So if there's more frequent stimulation, more stronger stimulation, you have more action potential, and you sense that. Okay. With phasic receptors, they're usually not active. Okay, here's the image of that. They're usually not active at rest. Okay, and then this provides information more about the intensity and rate of change of the stimulus. So when you see there's nothing, and then all of a sudden you have a burst of action, a burst of action potentials here, stimulation. Okay, you sense that change, that drastic change, and then it goes away when it stops. Okay, so you have bursts of action potentials here, and so you have that stimulus into the central nervous system, and then it stops when the stimulus stops and the intensity changes back down. So that becomes important when we look at the next thing, adaptation, and then looking into the specific receptor types. So adaptation, we talked about central adaptation with the sense of smell, how that adapts really quickly, okay, and how the nuclei influence the nuclei and the central nervous system influence your sensory pathway to reduce the amount of information that reaches the area it needs to in the central nervous system. Okay. So we talked about that and its effect on your reticular activating system. So your reticular activating system, again, we talked about last time how it wakes you up from in a sleep state. It'll actually wake you up at the right amount of stimulus, but it also brings your attention and awareness to different arriving stimulus. So at a certain point, that central adaptation will turn off your awareness to different things. Okay, so it turns off that the reticular activating system, just changes the amount of stimulus and sensation you're getting into your cortex, so you are no longer aware of it. So when you adapt to smell, this is the reticular activating system turning off that sensation in your mind, in your cortex, to where you're not receiving that anymore and you're not aware of the certain smell in the room until you leave and come back, or until a different smell presents itself. Okay, so that's central adaptation. With peripheral adaptation, this occurs in the peripheral nervous system at the receptor level. Okay, so this is where we're talking about the phasic receptors and the tonic receptors. So the phasic receptors have a faster adaptation. Okay, so this adapts fairly quickly to the stimulus. So we call it fast adapting receptors. The tonic receptors show little peripheral adaptation. Okay, so it doesn't, or it's slower at adapting. So this is an example of tonic receptors we'll see in a second is nociception, it's pain. So you don't adapt to pain very quickly, if at all. Okay. So now we're going to get into the specific receptor types. Okay, these different receptors, again, have a specificity. So nociceptive fibers sense pain. We have two different fiber types, type A and type C, and they sense different types of pain. Okay, so nociceptors are tonic receptors. Okay, we just talked about that, how the tonic receptors have little peripheral adaptation. Okay, you can have central adaptation with nociception, but very little peripheral adaptation. Okay. So with the nociceptors, they're present in your skin and joint capsules and the periostea of bone, so that outer layer of the bone, blood vessels, things like that. And then you have free nerve endings with large receptive fields that measure, can receive stimulus from temperature extremes, mechanical damage, or dissolved chemicals. Okay, so we have two different types of fibers. We have type A fiber and a type C fiber. Type A fiber is fast pain, so this is myelinated. Type A is fast. Okay, this is like pricking pain. So when you get like a prick or a cut or an injection or something like that, this sharp, quick pain that reaches the central nervous system quickly. It can sometimes trigger a reflex, so like stepping on a tack or something, you get that sharp pain and you get that withdrawal reflex or the flexor reflex. So it can trigger a reflex, and these fibers also allow for localization, pretty good localization, whereas type C fibers do not allow for a good localization. This is more dull, achy, burning pain, more generalized pain, excuse me. More generalized pain. This is slower pain, so it's unmyelinated. So the two fiber types are slightly different in the type of pain they sense and how quickly they sense it. The neurons that bring the sensation into the central nervous system release glutamate or substance P. And we talked about that with the endorphins and enkephalins earlier in the neurology modules, about how endorphins and enkephalins reduce the level of pain, specifically by reducing substance P. So different receptor types here. We have thermoreceptors and mechanoreceptors. So we're going to talk about the three different types of mechanoreceptors in a minute. But thermoreceptors sense temperature. Okay, we're more sensitive to cold temperatures than to warm, 3 to 4 times more sensitive to cold. These are carried on the same pathways as our pain sensation. Okay, and these are phasic receptors. So these adapt. These have peripheral adaptation fairly quickly. Okay, so we can walk into a room that's kind of cool, but we adapt pretty quickly. Okay, we walk in and we're like, oh, it's so cold in here. Then we adapt fairly quickly. We're not sitting there shivering. We just adapt. So that's thermoreceptors. Mechanoreceptors that is sensitive to physical stimuli, so distortion of the plasma membranes. So when we have this distortion of the cells, these get stimulated and open mechanically gated ion channels. So 3 different types of this: tactile receptors, so those on the skin, baroreceptors, those are internal. We'll talk about baroreceptors and chemoreceptors, and then proprioceptors, which we know a little bit about. That's in our joints. It helps us to sense where we are in space. Okay, so we'll talk about both of those. We'll start with the tactile receptors. Okay, we've gone over this before. We've seen this slide before. We all of these in the skin, so these feel touch. Okay, we have our free nerve endings, our root hair plexus, the tactile discs or Merkel cells, Ruffini capsule, laminated corpuscles, and the tactile corpuscles. So the free nerve endings are sensitive to touch and pressure, and these are tonic receptors. So they have smaller receptive fields, and they don't adapt. They don't have that peripheral adaptation as quickly. Okay, root hair plexus, these help to sense the movement of the hair. So when we have something that brushes across our body's surface, it moves those hair cells, or the hair follicles, and stimulates those nerves around the root hair. So it senses movements across our body surface. Then we have tactile discs. This is in that stratum basale, which we've already learned about in the epidermis, and it's sensitive tonic receptors. Okay, again, tonic receptors do not have peripheral or have very slow peripheral adaptation. And these tactile discs help to sense shape and texture. They deal with fine touch and pressure. Okay, and then we have the Ruffini corpuscles, also called bulbous corpuscles. These are sensitive to pressure and distortion. Okay, it's in the lower layer, the reticular dermis. Okay, so you have to have pretty deep distortion and pressure into the skin for these to be stimulated. Then you have Pacinian corpuscles, the lamellar corpuscles. They are deeper in the dermis. They sense deep pressure and high frequency vibration, and these are fast adapting receptors. Okay, so they have peripheral adaptation quickly. And then you have the Meissner corpuscles, which adapt within a few seconds, but these are higher up in the papillary layer of the dermis, and they're for fine touch and low frequency vibration. Okay, so there's the picture. You can go over what those look like again. Helpful for lab as well. Okay, so proprioceptors, we've learned about these before. Somatic sensation in joints, tendons, and muscles. We have the muscle spindles, and that monitor muscle stretch. Okay, so these are what we talked about with the stretch reflex, like the patellar reflex. So the muscle spindles sense that quick stretch in the muscle. The Golgi tendon organs, we talked about this too a little bit in that same reflex PowerPoint, where these are in the tendons, and they sense the tension within the tendons. So if the tendons become stretched too quickly, it will relax the muscle, so the tendon doesn't rip. Then we have receptors within the joint capsule itself. It can measure pressure, tension, and movement in the joint. Okay, and then we have baroreceptors and chemoreceptors. So chemoreceptors, they measure the concentrations of certain chemicals in our blood. So we have them in specific areas in our carotid artery and in our aortic artery, and measures pH, carbon dioxide, oxygen. Okay, these are all non-conscious things. We don't have to think about the concentration in our blood or in our different tissues of what our pH is and what our oxygen level is. Okay, our body just adapts to that based on these receptors. And then we have baroreceptors. Baroreceptors are throughout the body. Okay, we have them in our digestive tract, in our bladder, in our arteries, in our lungs. So it measures stretch and pressure in the organ. Okay, so they're free nerve endings, and there's some within our carotid artery, some in our aortic artery, in our sinuses, in those arteries. So it can monitor blood pressure, It's in our digestive tract, so it monitors different reflexes within the digestive tract, in our colon to trigger the defecation reflex. It's in our bladder to trigger the urination reflex. So the different receptors sense pressure and stretch within that tissue internally. Okay. Now we're going to talk specifically about the different sensory tracts that we have. Okay, we have 3 different ones we're going to talk about, but first we're going to talk about the organization of neurons. Okay, so we have 3 different order neurons. Okay, so we start from the periphery. Okay, we start more peripheral, then we come more central. Okay, so the first order neuron, this is most peripheral. So this is the neuron associated with the actual receptor cells. Okay, so that's our first order neuron, and it brings the information into the central nervous system. Okay, all that information comes up and through the dorsal root ganglion into the central nervous system, into the spinal cord. Okay, and then we synapse at the posterior horn. Remember, we have the somatic and visceral nuclei within the posterior horn, and so we synapse there, and when that synapse, we start the second order neuron. A second order neuron takes the information up to the thalamus. So we synapse here in the posterior horn, we go to a specific track, okay, a posterior column pathway or a spinothalamic pathway or the spinocerebellar pathway, and we go up. Okay, so these white tracks take us up, okay, they go up to the thalamus where we synapse again. And once we synapse the next neuron at the thalamus, then we start the third order neuron. And then from the thalamus, that neuron then takes the information to the specific area of the cortex or cerebellum. Okay, so we start more peripheral, and then we get more central. So the first order starts at this sensory receptor and goes to the spinal cord. We synapse to the second order neuron from the spinal cord to the thalamus. Then we synapse from the thalamus to the cortex. Okay. Along these pathways, at some point, we have a decussation. Okay. The second order neuron is responsible for the decussation. Either it happens here at that spinal level or it happens at the medulla. Okay. This decussation is the crossing over of that information to the opposite side of the central nervous system. And we'll look at that. It occurs at different points in some of these pathways. So we'll look at that. So we're going to talk about sensory pathways first. And the 3 we're going to do is the spinothalamic pathway, posterior column pathway, and the spinocerebellar pathway. So the spinothalamic pathway monitors for crude touch. It carries crude touch, pressure, pain, and temperature. Okay, the synapses in the ventral nucleus of the thalamus. Okay, so we see here that it decussates at the spinal level. And we have the pain, pressure, crude touch coming in, and it synapses right away, okay, and goes up the anterior column up to the ventral nuclei in the thalamus, and then goes to the primary somatosensory cortex. Okay, this primary somatosensory cortex is that post-central gyrus, okay, in the parietal lobe. And within the primary sensory cortex, we have this is what we call the homunculus, okay, where we have this depiction of the amount of innervation, amount of central nervous system that is delineated for the specific body part. Okay, so we have a lot of neurons in our central nervous system for our hands and our lips and our tongue, to be more specific with what we're actually feeling, what the sensation is, okay, as opposed to our back. Okay, our back, our leg, or our foot don't have as much area in the central nervous system dedicated to it. So we're going to have less specificity with what we're actually feeling. Okay, so this depiction of the different sized body parts based on how much cortical space is dedicated to it is called the homunculus. So we have our sensory homunculus, and then we also have a motor homunculus for our primary somatomotor cortex. So this pathway here is responsible for our phantom limb syndrome. Okay, remember that when we stimulate that specific track Anywhere along this track, we have that same sensation. So if we are missing a foot, but we stimulate at our dorsal root, that same neural path that would carry information for our foot, then when we have that stimulation, we have that sensation up in our cortex, that something is happening with our foot, or that we have a foot when we really don't. Okay. So that's the phantom limb syndrome that's carried by the spinal thalamic pathway. And then another thing that occurs with this spinal thalamic pathway is referred pain. So this is feeling pain in an uninjured part of the body when the pain is actually originating in another location. So we have a strong sensation of pain with organs that are innervated at the same spinal level. Okay, so we have an image of that here. So there's specific areas where pain refers. So our stomach refers pain here into the center here, with small intestine refers here, our large intestine is here. Okay, our ureters, you know, if you have that kidney stone that's working its way down, you have that pain around your back and flank down into your groin. Okay, this is all because these areas of the body are innervated at the same level as the organ. Okay, so when we have this pain here in the stomach, that's because the stomach is stimulating the nervous system at the spinal level. So it runs up that same track, and we have that sensation of pain here in the center, where the true pain is actually in our stomach. Okay. So referred pain and phantom limb syndrome is associated with the spinal thalamic pathway, which monitors for crude touch, pressure, pain, and temperature. Okay, so the next one we'll talk about is the posterior column pathway and then the spinal cerebellar pathway. They're both on this slide here. So the first posterior column pathway is on the side. We have two parts of that. Okay, this pathway carries information for fine touch, vibration, pressure, proprioception. And we have two different tracks, the fasciculus gracilis and the fasciculus cuneate. And these carry information from different parts of the body. So the gracile fasciculus carries sensations from the inferior half of the body, and it synapses in the medulla. And then the cuneate fasciculus is from the upper superior half of the body, the trunk, upper limbs, and neck. So both of those carry information for fine touch, vibration, pressure, and proprioception. So those synapse in the medulla, or those decussate in the medulla, they synapse in the gracile nucleus, at least the gracile fasciculus synapses in the gracile nucleus, and then it decussates over to the other side. It crosses over in the medulla, and then goes up the medial lemniscus into the ventral nuclei of the thalamus, the somatosensory cortex. Okay. And that, again, is where we have our sensory homunculus. And then for the cerebellar pathway, spinal cerebellar pathway, this is, again, our cerebellum is for controlling fine movements, corrective movements, posture, things like that. So we get information from our muscles, tendons, and joints into the cerebellum. So that's what's carried on that specific track to the cerebellum. It's again non-conscious, and it goes either through the posterior track or the anterior track. And this arrives to the Purkinje cells in the cerebellar cortex. And so it's more on the lateral side here, this yellow portion. And we have information that comes in and goes up on the same side, or crosses over and goes up. Okay. So we have proprioception of input from Golgi tendon organs, muscle spindles, joint capsules, like that. So we have information about where we are in space, where our joints are in space, to help with posture, movement, and corrective movements as well in the cerebellum. Okay, whoa, that's blurry. So this, I updated sort of one of the slides here because we were missing one of the tracks from from the other slideshow that you guys had. I guess I couldn't get it clearer than this, but there's a chart in your book that's similar to this, table 15.1, that has all the sensory tracks, and it shows you what it senses, the first, second, and third order neurons, where those are and where they go, the final destination, okay, is where they go, and when they cross over. So some of them cross over at the specific spinal level. So this one, axon second order neurons, cross over at the level of entry. And then this one with the fasciculus gracilis, it crosses over in the medulla before joining the medial lemniscus. Okay, so you can look through that, and it helps get you kind of an all-in-one little chart to compare and contrast the different sensory tracks. So now we're going to talk about motor tracks. Okay, so we have upper motor neuron and lower motor neuron that both are the efferent division, okay, from the central nervous system down to the peripheral muscle, skeletal muscle. Okay, and we're going to talk about the corticospinal tract, corticospinal pathway, lateral pathway, and medial pathway, which is made up of a couple of different pathways here. Okay, so we'll start with the corticospinal tract. Okay. This begins in the pyramidal. This is a pyramidal system, so that it begins in the pyramidal cells in the cerebral cortex, and this controls voluntary muscle movement. So there are 3 descending tracks. So we start with the corticobulbar tract. So we start in the pyramidal cells here, and we go down into the corticobulbar tract. Okay. When we get down to the medulla, 85% of that crosses over, decussates at the medulla, and goes down to the opposite side. When it goes down, after it crosses over, it becomes a lateral corticospinal tract. There are 15% that stays on that same side, goes down the anterior corticospinal tract before it decussates at the spinal level. Okay, so we have our corticobulbar tract. Then that splits, 85% crosses over to the lateral corticobulbar tract, corticospinal tract, and then 15% stays on the same side for the anterior corticospinal tract, and then that decussates at the spinal level to go out to the spinal nerve, or the skeletal muscle. So the corticobulbar tract, you see, also has a few things that come off, a few neurons that come off. These go, these neurons from the corticobulbar tract synapse at the motor nuclei for cranial nerves. Okay, so the different cranial nerves, we list them here, 3, 4, 6, 7, 9, 11, 12. So it synapses at the nuclei for those, and then goes to the skeletal muscle. So it control eye movements, facial movements, head and neck movements, tongue movements, muscles of the throat. So all of these control muscles. So motor, these are motor responsible for skeletal muscle movement. Okay, so that comes directly off that corticobulbar tract. And then you have the corticospinal tracts, lateral and anterior. And then again, we talked about the homunculus. This is the motor homunculus. So it's similar to the sensory, okay, but it's a little bit different depending on how the muscles are used, how fine-tuned you need it. So the hand may be a bit bigger, more tongue movement, so the tongue is bigger in the motor one. Okay. All right, now we're going to talk about medial and lateral pathways. So the medial pathway is broken up into vestibular tract, tectospinal tract, and medial and lateral reticulospinal tracts. So these medial and lateral tracts are extrapyramidal, so they're outside they don't start with the pyramidal cells. Okay, in the cortex, like the corticospinal tract starts in the pyramidal cells in the cerebral cortex. Okay, these start elsewhere. Okay. So these can help modify skeletal muscle contractions and movements and facilitate or inhibit lower motor neurons. Okay, so medial pathways, these control muscle tone and gross movement of the head and neck and proximal limbs. Okay, so medial pathway is muscle tone of the trunk, neck, and proximal limbs. So it's more medial. then we have the lateral pathway. Lateral pathway controls muscle tones, precise movements of distal parts of the limb. Okay, so that's more lateral. So the lateral pathway controls more distal movements, and the medial controls more proximal movements and muscle tone. So we have the vestibulospinal tract. This primary goal is for posture. The vestibulospinal tract here is concerned with posture and balance. It also sends motor commands to alter head, neck, eye movements, things like that to maintain proper head and neck positioning. You have tectospinal tracts. These are nuclei within the superior and inferior colliculus. So we talked about these before. Superior and inferior colliculus has the visual and auditory reflexes. So we have direct changes with head and neck upper limb positioning due to bright lights, sudden movements, loud noises as a direct reflex from the superior and inferior colliculus. So that's the tectospinal tract. And we call these the superior and inferior colliculi the tectum. Okay, these two colliculi are called the tectum, so the tectospinal tract. May help you remember that. Then we have the medial and lateral reticulospinal tracts. Okay, this is concerned with the reticular formation. Okay, the medial is is ponto reticulo, so it's in the pons, and the lateral is the medullary, so that's in the medulla. Okay, so it starts in the pons and goes to the reticular, the part of the reticular formation in the pons, and part of the lateral has to do with the part of the reticular formation in the medulla. Okay, and these control these control our movements related to our awareness to different stimuli. Okay, so this directs movements to specific stimuli. And again, all of these are part of the medial pathway, so it controls the movements of more medial structures, so head, trunk, proximal limbs. Okay. Now, the lateral pathway is controlling more distal movements, so the rubrospinal tract starts in the red nucleus. Rubrospinal tract is the red nucleus, okay, and this monitors distal muscles of upper limbs. Okay, so when we're moving our arms, and it helps coordinate that muscle movement. The basal nuclei and the cerebellum, these respond these get feedback from muscle contractions, and so this is corrective movements, also coordinating complex movements. And the basal nuclei helps to coordinate like things like walking and running. The more complex pattern of movements, it maintains background tone within these cycles of movement. Okay, and the cerebellum helps to monitor our body's position based on proprioception, visual, and vestibular inputs. Okay. So those basal nuclei and cerebellum. Okay, so that is the end of the sensory motor pathways. So that it was a lot of information, but look over each of the tracks, make sure you know what information they carry, where they go, okay, where they are on the spinal cord, where they decussate, things like that. We'll go over a review for the exam, or I'll do another video going over the review. The review is posted on Canvas, so you should be able to download that and start marking off things and going through the PowerPoints to make sure that you know the information that's on the review, and then you can watch the episode, the podcast that I do for going over that specific sheet. So I'll go over that sheet and I'll have the different PowerPoints up and talk through each of the points that you need to know for the exam. So.